Hi everyone, welcome to my channel, Interview Questions and Answers for a Quality Control. What is quality control? The term quality control refers to the sum of all procedures undertaken to ensure the identity and purity of a particular pharmaceutical product. It involves in chemical, physical and sometimes microbiological testing of a pharmaceutical product. Quality control involves testing of pharmaceutical products against the specifications. The other responsibilities of quality control are sampling of raw and packing material, testing of raw material, packing material, in-process, finished product and stability batches, sampling and testing of water, calibration of instruments, preparation of specification of raw, packing, in-process and finished products, preparation of standard test procedure of raw, packing, in-process and finished products and reporting of result after analysis and preparation of COA. What is calibration? The demonstration that a particular instrument or device produces results within specified limits by comparison with those produced by a traceable standard over an appropriate range of measurements. What is qualification? The action of proving that any equipment or process work correctly and consistently and produces the expected result. Qualification is part of, but not limited to a validation process, installation qualification, operation qualification and performance qualification, the act of planning, carrying out and recording the results of tests on equipment to confirm its capabilities and to demonstrate that it will perform consistently as intended use and against predefined specification. What is change control? It is an approved procedure which is taken to change in any documents, standard operating procedures, specification, process parameters and change in batch size etc. Change control is raised by user department as per requirement and finally the change control is approved by quality assurance. Change control can be raised through software or through manually. What is chromatography? Chromatography is a laboratory technique for the separation of a mixture. The mixture is dissolved in a fluid called the mobile phase, which carries it through a structure holding another material called the stationary phase, and the separation is based on differential partitioning between the mobile and stationary phases. What is HPLC? High-performance liquid chromatography is a technique in analytical chemistry which is used to separate, identify, and evaluate each component in a mixture. What is system suitability? Before start of analysis of the chromatographic system like HPLC GC system suitability has to perform to know that the system is working properly or to know the performance. System suitability criteria may include such factors as plate count, tailing, retention, and slash or resolution in the above factors are most important as they indicate system specificity, precision, and column stability. What is trailing factor? The tailing factor is a measure of peak tailing. It is defined as the distance from the front slope of the peak to the back slope divided by twice the distance from the center line of the peak to the front slope, with all measurements made at 5% of the maximum peak height. What are working standards? Working standards are defined as the samples of a drug or substance that are used as a reference during the manufacturing process. These standards are prepared by the manufacturer and are typically calibrated against a reference standard. The purpose of working standards is to ensure that the manufactured product is consistent and meets certain specifications. Working standards are used to measure the quality of a product during its production and are typically used in the laboratory. What are reference standards? Reference standards, on the other hand, are highly purified samples of drugs or substances that are used to establish the identity, strength, quality, and purity of a manufactured product. Reference standards are usually obtained from a recognized source, such as a pharmacopoeia, and are used to verify the accuracy of working standards. The use of reference standards ensures that the manufactured product meets the standards set by regulatory authorities and the pharmacopoeia. What is a dissolution test? Dissolution is the process in which a substance forms a solution. Dissolution testing measures the extent and rate of solution formation from a dosage form, such as tablet, capsule, ointment, etc. The dissolution of a drug is important for its bioavailability and therapeutic effectiveness. What is Q stands for in dissolution? Q is the amount of dissolved active ingredient specified in the monograph which is required to be released in the stated time, expressed as a percentage of labeled strength. Then the batch of the tablet or capsules is acceptable, if each unit is not less than Q 5%. Which tablets are used in calibration of dissolution apparatus, non-disintegrating, salicylic acid, and disintegrating, prednisone. Tablets are used in the calibration of dissolution test apparatus. What is Karl-Fischer titration? Karl-Fischer titration is a classic titration method in chemical analysis that uses coulometric or volumetric titration to determine trace amounts of water in a sample. What is stability study? Stability studies are performed in life sciences, chemical, and food and beverage industries to determine the effects of environmental conditions on product quality. Environmental conditions can impact product shelf life, and the viability of product formulation. In stability, how many conditions are there? There are 3 stability conditions: long-term or controlled room temperature, accelerated and intermediate. What is the ultraviolet and visible spectroscopy range? The range of UV spectroscopy is 200 to 400 nanometers, and visible spectroscopy ranges from 400 to 800 nanometers. What is audit trail? Audit trail is nothing but secure, time-stamped, computer-generated electronic records that allows for reconstruction of the course of events relating to the creation, modification or deletion of electronic records. It is a chronological record of the activity that happened, such as what, when, why, where, etc. What is 21 CFR Part 11? FDA 21 CFR Part 11 represents electronic signature and electronic records. What is analytical method validation? Method validation is the process used to confirm that the analytical procedure employed for a specific test is suitable for its intended use. Which test performed in analytical method validation? Specificity, system suitability, system precision, method precision, intermediate precision, robustness, accuracy, linearity and range, stability, solution, limit of detection, limit of quantitation. What is infrared spectroscopy? Infrared spectroscopy is a technique used to study and identify chemical substances by measuring their interaction with infrared radiation. This interaction can occur through absorption, emission, or reflection of infrared light. Infrared spectroscopy is valuable for analyzing the functional groups and chemical bonds present in solid, liquid, or gaseous samples. What is 5-point pH calibration? A 5-point pH calibration is a process where a pH measuring instrument is calibrated using standard buffer solutions at 5 different pH values. In this case, the calibration is performed using buffers with pH values of 1.68, 5.01/5.00, 7.00/7.01, 10.00/10. 01, and 12.45. This calibration ensures the accuracy and reliability of the pH measurements taken by the instrument across a wide pH range. What is pharmacopoeia? A pharmacopoeia is an official and legally binding collection of standards and quality specifications for medicines used within a specific country or region. It provides guidelines for the quality, purity, strength, and labeling of pharmaceutical substances and dosage forms. Pharmacopoeias are crucial for ensuring the safety and efficacy of medicines. What are the types of pharmacopoeias? United States Pharmacopoeia, British Pharmacopoeia, European Pharmacopoeia, International Pharmacopoeia, Japanese Pharmacopoeia, Indian Pharmacopoeia, Chinese Pharmacopoeia. I hope you liked the video. Thank you for watching.